Hello students, so welcome again. We are going discussing about the chapter 6, seed dormancy and the germination. In the last few parts of the chapter we discussed about the seed dormancy's definition, its type, then seed germination definition and its type, seed germination, then methods of seed breaking. Now today we'll discuss about the methods of seeds breaking, the second type, that is the stratification. So this stratification, it is a method of handling dormant seed in which the imbibed seeds are subjected to a period of chilling after ripen the embryo in alternate layers of sand or soil for a specific period. So it is known as the moist chilling, okay, or stratification process. It has a various uh, types. The first is the outdoor stratification. So if refrigerated storage facilities are not available, the outdoor stratification may be done either by otherwise storing seeds in open field conditions, in deep pits, or in the raised beds enclosed on wooden frames. However, it is likely that seeds are destroyed in outdoors by excessive rain or the freezing or the drying or by rodents. Okay, the seeds are placed in alternate layers of sand to provide an low temperature and proper aerations in the stratification pit. The top is covered with the sphagnum moss to maintain the moisture level. The pit or tray is aerated at regular intervals to maintain appropriate moisture status. Now the next, that is the refrigerated stratification. So it is the alternative to outdoor field stratification in refrigerated stratification. So it is useful for small seed lots or valuable seeds that requires special handling. So the dry seeds should be fully imbibed with the water prior to refrigerated stratification. So 12 to 24 hours of soaking at a warm temperature is sufficient for seeds without hard seed coats. So after soaking, seeds are usually placed in a convenient size box in alternate layers of well washed sand or pits, moss, or vermiculated area. So a good medium is a mixture of 1 part of coarse sand to 1 part of a pit, moistured and allowed to stand for 24 hours before use. So the seeds are placed in alternate layers of sand or the medium, we can say. So the usual stratification temperature is about 4 to 7 degrees Celsius. At a high temperature, seeds sprout prematurely and low temperature delays sprouting. So the medium should be remoistured. Okay, so the stratified seeds is separated from the medium prior to sowing in nursery beds. So the stratification of seeds results in quick and uniform germination, and therefore the seeds should be subjected to stratification invariably under all conditions. Okay, so this is the second part. Now the third is the leaching of inhibitors. So leaching means the soaking, keeping the seeds or the plant material for 20 more than 24 hours, we can say 12 or 24 hours, and that liquid form is known as a leachate one. Okay. So leaching of inhibitors. Some inhibitors and phenolic compounds are present in seeds covering of many species, which inhibits germinations. So therefore soaking of seeds in the running water for 12 to 24 hours or placing them in the water for few hours or more than 2 days help in leaching of the inhibitors, and phenolic compounds help in easy seed germination. So pre-chilling, next is the pre-chilling one. So the seeds of certain plant species dormancy can be overcome by pre-chilling treatment also. So in this treatment, the imbibeds, or we can say soaked seeds, are kept at a temperature of 5 to 10 degrees Celsius for, we can say, 5 to 7 days before sowing. After that, seeds can be sown in the field immediately. Okay. Now the next is the pre-drying method. So this is also a useful practice in some seeds to overcome seed dormancy. In this treatment, the dry seeds are subjected to a temperature of 37 to 40 degrees Celsius for about 5 to 7 days prior to sowing one. So after this, seeds can be sown in the field. Next is the seed priming. So in this, seed priming refers to the procedure followed by overcome dormancy in freshly harvested fruits. Most widely used seed priming procedures are osmo conditioning, infusions, and the fluid drilling one. So these are the three conditions. Now the next, that is the treatment with the chemical one. So some compounds other than hormones are also used to break dormancy. So thiourea is one example, known to stimulate germination in some kind of dormant seeds. The seeds are soaked in about 0.5 to 3% solution of thiourea for about 3 to 5 minutes. So afterwards, seeds are rinsed with the water and are sown in the field. Similarly, potassium nitrate and sodium hydrochloride also stimulate seed germination in many plant species. Okay, so the last one, that is a hormonal treatment. So among various hormones, gibberellic acid is commercially used for breaking seed dormancy in different types of seeds. So the concentration of gibberellic acid depends upon the kind of seed, but generally of a concentration of about 200 to 500 ppm is most widely used. So the cytokinin is another hormones used for breaking physiological dormancy and stimulating germination in seeds of many species. And next is the kinetin, and then benzyl aminopurine are commercial preparation of cytokinin used for breaking seed dormancy, and then soaking seeds in about, we can say, 100 ppm solution of kinetin for about we can say 5 minutes, which is highly effective for overcoming seed dormancy of many species. Again, there is the erythritol is there, which also stimulates germination in seeds of some species, which is a process within the endosperm. So thank you very much.